Hi, biology. I miss y'all. I have plenty of work for you to do. I'm in Milwaukee. I never can say that. It's right next to Lake Michigan. It is super cold. I got off the plane and I just thought I do not have the clothes for this weather. So definitely am ready to run back to the warm weather here. I have a lot of notes for you, and so I know that makes you really happy. I'm not going to make you copy down every single thing here, but you're going to copy down a whole lot of stuff. This is going to go quickly, but Miss Rhonda can pause it whenever you need her to pause it. Just make sure that if I say write something down, you've got it, because you know you're going to have a notebook test when I get back, and another notebook test, and then we're going to test probably the for real test over everything we've done on probably Wednesday when I get back. So get your little writing fingers ready. We are looking at active cellular transport this time. Notice that we do have a lot of key vocabulary. You don't have to write this down, okay? So do not write all these things down. But notice you've got this word here we've not really seen yet. Enumerate the 3 types of active cellular transport, and that's just, you know, saying list them off, and you know, number 1 is this, number 2 is that, number 3 is that. So that's maybe a new word for us. So you do need to copy most of this down, okay? We've got a lot of important phrases here. Active cellular transport. So please write pretty much this whole slide down. You're going to write this whole slide down. Yes, the whole slide. Active cellular transport are when particles move from less concentration to greater concentration, or against the concentration gradient. This is different from passive cellular transport. In this case, our arrow is going up, meaning that we're moving against the current, or we're climbing up the hill. Remember, passive transport, our arrow was pointing down. We did not need any energy. We were just simply falling off a cliff. Okay, but with active cellular transport, you have to have energy because you're going against the gradient from lower to higher. So it requires energy, and that energy is in the form of ATP. There are 3 types of active cellular transport. The first one is a molecular pump. We'll find that this is very important in our bodies. This goes on in every single cell over and over again. This is something that our bodies do that transmits signals, but it's very important. It involves the use of molecules to drive other particles in and out of the cell. So molecular pumps, we're going to be talking about those. The second type of active cellular transport, endocytosis. So please write that word endocytosis and circle the endo, E-N-D-O. And that means into. Okay, so into is endo, E-N-D-O. And then cyte, C-Y-T, is cell. I know that that's on one, well, probably on multiple tests, because, you know, cytoplasm is the plasm that's inside the cell. A cytology would be, you know, the study of cells. Cytoskeleton is the skeleton of the cell. So we see that a lot, okay? But endocytosis is moving something into the cell. You'll notice another new word here. The way that we get things in and out of the cell is with these little things called vesicles. And vesicles are just like little sacs that move in and out. There are some pictures here that you'll be able to see. The third way is when we're moving something outside of the cell. Okay, so exocytosis, write down that word. And then EXO means out, kind of like exit. And then cyte is cell. And then this osis is movement. I should have pointed that out whenever we studied osmosis. Okay, but endocytosis, movement into the cell. Exocytosis, movement out of the cell. And again, when we're doing exocytosis, just like endocytosis, we have to use these little sacs that are called vesicles. Okay, vesicles are these little sacs. I'm moving on, but y'all feel free to pause this when you need to. I know this is a lot. Molecular pumps. Now, with the molecular pump, again, we're moving the material up or against that concentration gradient. And you'll notice that what you've got here is this little, it's almost like a little, sort of like a little gate, but it's a protein that is going to open and close. Okay? There are different types of molecular pumps. Now, we do not need to know these types of molecular pumps here. This is not in our standards. Okay, so for example, when you have a primary active transport, this happened to be one of those extra credit questions that was on your quiz. We do not have to know this. This is not a part of our standard. So from this slide, all you need to write down is that these molecular pumps involves the use of other molecules to get things in and out of the cell. Okay? And they're pictured like a little gate. So you will be expected to look at something like this and notice that that is a molecular pump. Now, the most famous one that goes on in our body is this sodium-potassium pump. This goes on to transmit information in our body. It's called the sodium-potassium pump. All right, you do not need to write all of this down, but I do want you to write this last piece down. An example of that primary active transport is that sodium-potassium pump. It's found in nerve cells. So notice again, if you see pictures that look like this, okay, they look like these little gates that they open down at the bottom, towards the intracellular side, and then they release stuff up here at the top on the extra or outside of the cell. So it's kind of like a little Pac-Man. The mouth opens here and then it spits it out up here. Okay, these are molecular pumps. And the most famous one that you do need to know is that you've got a sodium-potassium pump in your nerve cells, and that's how our nerves transmit information. You do not need to know all this up here, but you do need to write down this that I have circled. Your secondary active transport, we don't need to write this down, but just be aware of it. An example is a hydrogen sucrose pump that is in our body that is going to allow these chemicals to go in and out of our cell. But again, we don't have to know that because it's a little bit above our standards, so don't worry about writing that. Okay, you do need to know these. Endocytosis. We already wrote down that it is movement into a cell with the use of vesicles. So make sure you have that word written down, vesicles. Now these are large molecules, so they're not small. So here's a word that you get to laugh at. It says the plasma membrane of the cell invaginates, forming a pocket around the particles. It touches off the pocket, which results into a new created intracellular vesicle formed in the plasma membrane. Okay, now that just means it forms a little sac called a vesicle to get the stuff outside, or excuse me, inside of the cell. Now there are 3 types of endocytosis. Now we do need to know some important stuff here that I need you to write down. Okay, number 1, phagocytosis, P-H-A-G-O. Okay, and then you're going to circle phago, P-H-A-G-O. Circle that. Phag, P-H-A-G, means to eat. Okay, so P-H-A-G is eating. Like we have an esophagus. Our food travels down our esophagus. Okay, Pinocytosis, P-I-N-O. We don't see this P-I-N-O very often, but you have to know the difference between these two. Phago means to eat. Just remember esophagus. You eat food, it travels down your esophagus. Okay, and this is the large particles taken in by the cell. These are your solids. Okay, Pino, P-I-N-O, cytosis. This is liquids. So Pino involves liquids. Now the only way that I can really remember that, or tell you to remember that, is that Pinot, there's a wine called Pinot Noir, and it's a liquid, so I just remember Pinot is liquid. I guess you could think of peeing being liquid. I don't know. Whatever works. Okay, then you've got receptor-mediated endocytosis. We do not need to know much about this. So you can just write down receptor-mediated endocytosis is an extremely selective process but we don't really need to worry about that one. So remember your big words from this slide. Vesicles, they're those little sacs that move things into the cell. Phagocytosis, solids taken into the cell. Pinocytosis, fluids into the cell. Here's some pictures that help us to understand. We got all kinds of vocabulary in here, but you got these little solid particles. This is phagocytosis. Notice all of these are endocytosis. This is the outside of the cell, the extracellular. This is the inside of the cell, the intracellular. Okay, notice cytoplasm, the plasm in the cell. So these little solid particles, the way that they get into our cells, the plasma membrane, this is that invagination process, that funny word that I know you're laughing at, so stop laughing. Be good. But it's wrapping around the little solid particle, and then it forms what's called a Faygo Zone. Here you go with that SOME again. And SOME, remember, is body. Okay, so this is like a little food body that's going into the cell. Okay, if you'll think about the article that you read yesterday, there was in the slide, there was something about pseudoscience. If you look at this pseudopodium, pseudo means false. Okay, so you saw that word pseudoscience or false science. Pod means foot. And so this is something that we'll see whenever we look at some different organisms and the way they move. They move with pseudopods, but or false feet. So this process of invagination, getting the solid food particle into the cell, these little pseudopods or false feet pop up right here. Okay, so we don't really need to know all of that, but we do need to know these words because we're going to see these words again. Okay, now, Pinocytosis. Again, we're getting these liquids in here. And then you've got this receptor-mediated endocytosis. When we talked about this being something that was very selective, notice the shape is important. Only certain shapes will get in there. So because something is receptor-mediated, it has little receptors here. And in this case, it's only letting the stars inside because the stars are the shape that these receptors are coded for. So the big takeaway from this is just to be able to recognize the drawing. And if you look at this, all of these things look like they take energy, and they involve these vesicles being pinched off. So if you see these little vesicles, you do not have passive transport, you've got active transport. Think of pinching off pieces of dough. That's kind of like what that is. It takes energy to do that. Exocytosis. We're sort of going in the opposite direction here. But notice we've got this organelle, Golgi Apparatus, in this picture. And this is something that we've talked about, and I know it's difficult to understand, but the function of that Golgi Apparatus, remember, is to take the proteins and things that the cell makes and then put the extra little pieces and parts on them. Just like when you get a car out of a factory, it doesn't have anything special on it. The dealership will put some extra special stuff in there to get you to buy the car, or maybe you order the car from the factory and you say, I want this stereo, or I want this color paint job. Well, that is what the Golgi Apparatus does for proteins. So these basic proteins are made by the cell. That Golgi Apparatus packages the proteins or modifies these proteins. Well, once the cell makes these proteins, it has to get them outside the cell to do whatever it is that these proteins are supposed to do. So, again, this takes energy. So the Golgi Apparatus is these little vesicles are being pinched off and moving outside of the cell. Just in case you did not get any of these things, make sure that you have this written down. Okay? Your active cellular transport is the movement of particles against, so your key word here, against, a concentration gradient. You've got 3 types. The molecular pump. Make sure you remember that the sodium and potassium pump is how our nerve transmissions are going from one nerve to the other. You've got endocytosis. Things are coming into the cell through these vesicles. And you've got exocytosis. Things are moving out of the cell through vesicles.